Hi, I'm Thomas Litsche, and in this video abstract I'm going to talk about our paper in the new Journal of Physics titled Freeze-thaw cycles induce content exchange between cell-sized lipid vesicles. In this work we wanted to find out more about how life could have emerged under prebiotically plausible conditions. We were looking into some physicochemical factors that might explain how information between early protocells could have been exchanged. Protocells are precursors of cells, and here we use lipid vesicles as model protocells. They're also called liposomes or GUVs, which stands for giant unilamellar vesicles. It is commonly believed that compartmentalization must have played an important role in the origin of life. Compartmentalization is a key principle for life in general, and it is likely that isolating chemical reactions in compartments was a prerequisite for the emergence of life. Another basic biological principle is exchange of information between compartments or individuals in general, as we also see in modern life, for example in more complex organisms as sexual reproduction. Here we wanted to investigate how protocells were able to exchange information, which means exchange some of their content. They did not have a complicated protein machinery like single-celled organisms like bacteria. So the question is, how could these simple precursors of cells that most likely did not contain any kind of protein have done that? It is likely that they relied on physicochemical factors, like environmental events, to do so, especially any kind of periodically reoccurring events. One parameter that generally oscillates over time is temperature. It does so, for example, on a daily cycle. Changes in temperature especially get interesting at around the freezing temperature of water. Generally, one scenario of where life could have emerged is in pools of water that periodically freeze and thaw. Unrelated to compartmentalization and content exchange, previous work has shown that in a system that undergoes freezing and thawing, certain chemical reactions important for the emergence of life are favored. In this work, we look into physicochemical advantages of such an environment that could have enabled an exchange of content between early protocells. So to look into content exchange between our model protocells, we generate two populations of lipid vesicles, and we freeze and thaw them. And what we see when we look at them after they fully thawed is that there is content mixing happening, and it is actually very thorough. But more about that later. One important part of this work was to develop a high-throughput method to study these vesicles under various environmental conditions and driving forces. In future studies, we want to test a large range of different parameters and different prebiotically plausible molecules. So to do so, instead of commonly used containers like microtest tubes, we use glass-bottom microtiter plates. These allow high throughput because we can compare many different conditions at the same time, one condition in each well of the plate. But especially the glass bottom of these compartments plays an important role in our protocol because it allows us to generate, freeze and thaw, and image our vesicles all in the same container without pipetting steps in between. We generate our vesicles through emulsion transfer. That means we first need to generate water and oil emulsions, stabilized by lipids. Because we want to study the exchange between two distinct populations of vesicles, we need two different kinds of emulsions. Here we use two solutions with different fluorescently labeled DNA. In the next step, we put these emulsions on top of a water-oil interface in our glass-bottom microtiter plate. Now we have a layer of oil which contains microdroplets on top of an aqueous layer beneath. To get lipid vesicles from these droplets, we put the entire microtiter plate in a centrifuge and spin the droplets through the water-oil interface. Here you can see a confocal microscope image of the liposomes we get from this process. And now, as I mentioned, we freeze and thaw these vesicles, and as a result, their contents mix. Even though similar behavior for the freezing and thawing of lipid vesicles has been shown before, our results differ quantitatively and qualitatively from previous work, as we point out in the paper. We further think the microtiter plate layout will enable efficient future screening experiments. We looked into a number of different conditions. Specifically, we varied freezing and thawing rate, and surprisingly found that content mixing depends on both the freezing and the thawing rate. We see the most efficient content mixing when we freeze our samples fast at -80 degrees Celsius and then slowly thaw them at room temperature. In the paper, you will find a detailed analysis of these experiments. The biggest surprise was that the mechanism which we found responsible for this content exchange was fundamentally different from previous reports for content exchange between vesicles. With these results, we hope to give a new insight in how basic environmental driving forces could have contributed to the emergence of life. To find out how exactly we think these vesicles exchange their content, and to see some other experiments and more detailed analysis, we invite you to take a closer look at our paper, which is available online and with open access in the new Journal of Physics.